Hello everybody! So we are finally on the last part of topic 17 and we're talking about phylogenetics and its role in conservation. So far we've only talked about conservation as a topic, but we're finally getting to the part where I want to talk a little bit about how phylogenies could be useful for making conservation decisions. I'm going to walk you through a couple of different metrics that have been proposed over the last couple of years. Some of them are fairly old and have been around since the early 1990s, but we'll talk about these because they have some really nice implications for what we do today. I will expect you to know how to interpret these metrics based on some simple examples. I will also be posting the papers, the original journal articles that refer to these metrics, and so please make sure that you take a peek at those just to get a general understanding of what is going on. So we talked a little bit about the history of conservation in the United States, and of course that was a very cursory overview. There's a lot more to discuss, but as long as you understand where we come from in the United States when it comes to conservation, you should be good to go. We also talked a little bit about different approaches to conservation, which included both species and broader approaches that focus more on ecosystems or especially hotspots. And the reason hotspots tend to be the focus of conservation is because they have a high number of endemic species. And if you remember, endemic species are those that are found in typically more restricted areas, smaller geographic ranges. Those rules don't always apply, but endemism is usually typically just described as species that live in a limited area. And then we talked about the IUCN categories that are found on the red list. So please make sure you know those categories because we're going to come right back to them at the very end of this topic. So let's get into this. First, we're going to talk a little bit about conservation and the goals of conservation from a phylogenetic point of view. If you recall, at the very end of the last video, I mentioned 3 basic goals or things that we try to approach when we're thinking about conservation from a typical traditional standpoint. And that would be to try to avoid or prevent against extinction, number 1. That is always the goal of conservation. Number 2, to try to prevent extinctions by conserving either areas of really high species diversity and species richness, more specifically. And then that also means that you should try to conserve or preserve as many species as possible. So those 3 things go together. How phylogeneticists might view this is a little bit different. So instead of focusing just on taxa or species, the idea is that from a phylogenetic viewpoint, we try to conserve as much evolutionary or phylogenetic history as possible. We also tend to focus more on evolutionary diversity. But basically the idea is that instead of focusing just on taxonomic diversity, we're focusing on diversity across the tree of life. And then finally, not just are we focused on preventing extinction of species, but we are attempting to prevent losses of branches on the tree of life. We are basically coming full circle and talking about the tree of life, which we discussed at the very beginning of the semester. And one caveat of that is that we don't really have a good grasp of a lot of taxonomic groups' evolutionary history, but we're getting close. So there have been numerous metrics proposed since the early 1990s. These metrics are basically ways for us to calculate diversity. So the origins of these metrics that I will be proposing was really just to help us figure out how to measure biodiversity. Do we measure it just in terms of looking at the number of species that live in an area, or do we consider other factors? So we're going to talk about some metrics that have proposed other factors, and because this is a systematics class, we will be talking about the factors that are related to phylogenies. So we'll talk about a couple of metrics. There's 3 major ones that I will introduce to you, and they're all based on one basic approach. Let's first talk about how we tend to see diversity on our planet. I'm just going to call this taxonomic diversity. This means diversity of taxa. We tend to measure diversity or biodiversity in an area based primarily on the species that are there or the taxonomic diversity that is there, right? Species diversity is the same as taxonomic diversity. So if you have an area, and you can think of really any area that you're looking at, the number of species tends to give you a good estimate of how much diversity is there. The number of species is usually defined as species richness. And this goes back to a concept that you learned early in your biology career. There is also another value which we won't really revisit again, but there's also something called species evenness, which is the relative proportion of all of the species in an area, the relative proportion or proportions of all the species in an area. And these two measures together, species richness and species evenness, so here we have the first definition, species richness and species evenness, together these equal what we now know as species diversity. So species diversity or taxonomic diversity today is measured as the number of species and how proportional those species are relative to one another. Okay, so this is how we tend to look at diversity in an area, but we are going to talk about metrics that don't just look at species diversity. So this taxonomic diversity is the traditional approach. Traditional approach to looking at diversity. We're going to explore a couple of different ones. So let's get into the nitty-gritty. What is it that we actually want to think about when we're thinking about phylogenetic diversity? Phylogenetic diversity is a relatively new concept, but it started as a topic of discussion in the early 1990s. Specifically in 1992, there was a paper published, and we'll talk a little bit about that paper, which was the foundation really for all of this work. And so this topic has been around for a while, and different measures have been proposed. Phylogenetic diversity is basically a measure of biodiversity, which is what we just talked about, but instead of just basing it on the number of species or species evenness, so species richness and species evenness, we also think about the phylogeny. It's a measure of biodiversity that is based on phylogeny. And this phylogenetic diversity measure was first proposed by Faith in 1992. So we will talk a little bit about that paper. I've broken down some of the concepts from that paper, which I will post on Moodle if you are interested in reading it yourself. And we're going to take these approaches that were proposed by Faith and explore them in a little bit more detail. So phylogenetic diversity is a measure of biodiversity, or just species diversity, based on the phylogeny itself. And the general idea here with phylogenetic diversity, like why we use it and why we consider using this instead of just taxonomic diversity, is that we want to measure not just the number of species or the species richness in an area, but we also want to measure or examine where those species are found in a phylogeny, because that makes a really big difference. And one of the hallmarks of phylogenetic diversity is that it considers branch lengths. And the reason for this is that a longer branch length tends to be an indicator of feature diversity. So if we assume, I'm going to draw just a branch length. So let's say here's a phylogeny of, say, 3 different species. If we convert this into a rectangular form, so we'll do A, B, and C into a rectangular form. Let's say that we've got A, and then here we've got B and C, which are sister taxa. The general idea is that the longer a branch is on a tree, and we know that there are There are exceptions to this rule because of long branch attraction. But if we assume that there are long branches, along those branches, right, the branch lengths will be an indicator of how many evolutionary changes have occurred on those branches. The shorter the branch lengths, the fewer number of evolutionary changes have occurred on that branch length. When we talk about phylogenetic diversity, everyone, we are really talking about molecular diversity. You can, if you want, think about this in terms of species A has built up numerous, say, physical features or behavioral features that make it unique and separate it from B and C, but we're going to think of this in terms of molecules. The branch lengths basically equal feature diversity. And feature diversity just means unique mutations that have built up over time. So branch length equals feature diversity. And feature diversity is really just, in our simple terms that I want you to think of for this class, is the number of unique features that have built up over time. Those features, think of them simply as mutations that could potentially result in new physical features, new adaptations to the environment, new behaviors, new morphology, etc., etc. So branch lengths are relatively important. Those species that have longer branch lengths, if the phylogeny has been done correctly and we've taken long branch attraction out of the picture, then those longer branches give us an indication of how unique perhaps that species is. So let's take a look at this simple example. If we imagine that we have two different phylogenies, the first one is a phylogeny of animals that you might recognize. These are ungulates. These are ungulates. So we'll just say ungulate phylogeny, ungulates, and then the second tree is just a generic animal phylogeny. So we'll just say these are animals, going all the way back to some of the invertebrates. Okay, so the invertebrates would be a grasshopper and a scallop, and then we've got a fish, an owl, and a rhino. Now, what is the difference between these two different trees? If we look at the ungulate tree, you will notice that there are various species that you might recognize. There is our horse, so we'll write some of these names on here. This is a zebra, or you might call it a zebra. This is a generic rhino. Here is a goat, and we've got a cow or some bovine animal. So we have a tree that represents various sister relationships, horses and zebras, and then we've got rhinos, which is a sister to the equines, and then we have goats and cows that share a sister relationship. You will also notice that the branches here are relatively short, and the reason for that, especially if you look at the horse and zebra, and then the branch that leads them to their relationship with rhinos, is that those short branches represent really either short evolutionary history time or the number of, say, mutations that have built up along those branches. The general idea is that the longer the branch, the longer the evolutionary time, which also gives evolution a chance to build up numerous mutations over time. And it also depends on the genes that you're looking at. If we now compare the ungulate tree to the generic animal tree, and we go back to our animals, so here we have, again, a generic rhino. We've got an owl, one of my favorites. We've got some fish. We've got a scallop, and we have a grasshopper. What you will notice in this tree, and this is partly because we're looking at animal groups that have evolved over long periods of time, there are some branches that are really, really long. And then there are other branches that are a little bit shorter. But the general idea is that if you trace the branch lengths from invertebrates all the way to where the vertebrates are, those branch lengths are really long. And fish have been developing over time, separate of all of the other vertebrates, building up different mutations depending on the fish groups that you're talking about, separate from the rhino and owl group. What really is this suggesting? Which tree has the highest phylogenetic diversity? And how you might simply look at this is if you are only looking at ungulates, which is a small group of vertebrates, right? Ungulates are also a small group of mammals. There is very little phylogenetic diversity here. If you compare that to the animal tree, you have a larger variety, a wider variety of different animals in that tree, plus the branch lengths are longer, which are suggestive of unique features and mutations that have built up over time. So comparing these two different trees gives us two very different ideas of evolution. Ungulates compared to animals as a whole are less phylogenetically diverse in terms of the phylogeny that we have included. So if we imagine that ungulates as a whole goes extinct, let's just say all of them go extinct, and you compare that to the animal phylogeny, ungulates is represented by the rhino in the animal phylogeny. So let's say that in this tree, we suppose that that entire clade, the ungulate clade, goes extinct, but in the animal phylogeny, rhinos, which is representative of all of the ungulates, goes extinct. We still have owls and fish and scallops and grasshoppers. So what this is basically suggesting is that ungulates is lower in phylogenetic diversity, whereas animals, just this tree in this particular examples, has a higher phylogenetic diversity. So that really is just a measure of how many different phylogenetic groups or taxonomic groups are found in that tree, and this also gives you a sense of which kind of groups we should focus our conservation efforts on, right? Now, this doesn't really work that simply for this particular example, but it gives you an idea. So longer branch lengths give us unique features over time, and shorter branch lengths really don't. So let's keep that idea in mind. Another way to look at this is if we have this phylogeny on the left, which I've drawn in a diagonal up shape this time, and we imagine that all the blue lineages, the light blue color, these are extinct lineages. So all of these blue lines in the phylogeny, these have gone extinct. Okay, and if you count these up, we have a total of 6 different extinctions. There are 6 extinct taxa, whatever those taxa might be. Now let's imagine that in a different universe, we have the same phylogeny, and we also have 6 taxa that have gone extinct. Just to count these up, 1, 2, 3, 4, 5, 6. This is also 6 extinct taxa. But what is the big difference between these two trees? So these are just two hypothetical situations, right? Both on the left and the right, we've got 6 extinct taxa. But which tree has lost the most phylogenetic diversity? If these two trees are looking at the same number of taxa and the same taxonomic groups, you will recognize that the phylogenies are the same, right? So these are the same phylogeny. The same phylogeny and the same topology. So the question here is, which is more phylogenetically diverse if we compare the two trees relative to what is lost over time? If you, by chance, through evolutionary time, lost these species, so this is let's just say that this is species A, and this is Z, and this is C, and this is E, and this is G, and this is J. That's J. Okay, and here we've got species B, and this is A, and species Z, and we've got C over here, and let's say this is D, and this is E, and then we leave the rest alone. In which case is the largest phylogenetic diversity saved? So think about this for a moment, and you will notice that if, let's say, these 3 long branches go extinct with all of these terminal taxa here at the end of these branches, you are losing a huge amount of phylogenetic diversity. You are really just preserving this last little clade. And this clade might all be species that are very similar. Species that share a recent common ancestor but that don't have a lot of feature diversity. Let's say that all of these other branches are different reptiles, and all that we've saved here, all that remains after extinction events, perhaps caused by humans, are, say, a group of birds, but we've gotten rid of all the snakes and the lizards and all of the alligators and other crocodilians, and we've gotten rid of all of the related amphibians, say. That is pretty devastating. Compare that to the tree on the left, where you're losing short little branches of taxa. Yes, it is still devastating that you've lost these taxa due to extinction, and here you've lost a relatively long branch as well, but you haven't gotten rid of huge swaths of phylogenetic diversity in that tree. So the idea is that it makes more sense to focus conservation efforts on phylogenies if you can focus your conservation efforts on preserving the highest amount of phylogenetic diversity. So we will talk about measures that do this here in a little bit. So here's another way of looking at this. The measure that is often used for phylogenetic diversity is the number of changes along a branch. And this is an oversimplified example, but it gives you finally another idea of how this works. Let's say that we can just call these species 10, 8, 6, and 2, and the other species on the tree have gone extinct. But 10, 8, 6, and 2 have remained. So all the other branches are gone. And then I've got a random species number 4 on there, which I will talk about here soon. 2, 6, 8, and 10, which are the species highlighted here, had the maximum expected phylogenetic diversity in this tree because they have the most evolutionary changes over time. They have the longest branches. And those branches, the branch lengths, are really important. They have the longest branches. In other words, the most feature diversity. If we got rid of species 2, for example, which by itself on its branch alone has 1, 2, 3, 4, 5, 6, 7 different unique evolutionary instances that have occurred over time. So if you got rid of species 2 or species 2 went extinct, you've lost that entire lineage of uniqueness. All right, if we look at the phylogenetic diversity for number 2, it is actually counted as 12. Now, how is that possible? That is because the branch that extends below it actually adds some additional steps. So we've got 8, 9, 10, 11, 12. If we got rid of species 2 or species 2 went extinct, this entire line of evolutionary changes that led up to species 2 will be lost. The phylogenetic diversity score for number 6 is 10. And here you can count these up again. So we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 different unique evolutionary instances. The PD for 8 would be 11, and you can count those up yourself. Make sure that you can double check and calculate these simple measures and values. And the PD for number 10 is 4. The PD for the total tree that we have here is 28. There are, in combination with all the species that we are focusing on, which are 10, 8, 6, and 2, and let's just imagine that all the other species went extinct, the total phylogenetic diversity would be 28. What would happen if, let's say that we have a species 4 that is still living? So now let's imagine that, I'm going to mark these in blue, let's imagine that these branches that I am marking with an X have gone extinct. Species 4 is still there, it's still living, but it is an endemic species. If you recall, I mentioned that we tend to focus a lot of our conservation efforts on endemic species. If taxon 4 is endemic, should it be given conservation priority? So let's say that we have species 10, 8, 6, and 2, as well as 4, and we have to choose which one should get conservation priority. For which one should we come up with some conservation plans, measures, public education, etc., etc.? If you compare 4 to the other species that are there, it only has 4 unique evolutionary changes, and really just 1 on its own branch. If you got rid of number 4, and let's say number 4 went extinct, so let's say this one is gone. Number 4 is gone and it's gone extinct. Really what you've lost is only 1 unique evolutionary change. So think about that, right? Phylogenetic diversity is important because it focuses on species that have over time evolved potentially unique behaviors, morphology, and other features that make them very different from other species, and which might suggest that we should focus our conservation efforts on those particular species because they are an emblem of phylogenetic diversity. So the answer here for taxon 4 is no. If taxon 4 goes extinct, you're really only losing 1 unique feature or molecular change along that branch. What would be really sad is if you lost species 2, or even if you lost species 8, and the worst of all is if you lost all of these species. Let's say that we decided to save species 4 because it's endemic, it's some really cool lemur that lives in Madagascar, but we've got species 10, 8, 6, and 2, which are not really of conservation concern. They're not perhaps on the red list. What if we got rid of branches 10, 8, 6, and 2? We would lose all of that phylogenetic diversity just to save an endemic species that it really isn't that phylogenetically unique. So hopefully you understand this idea of phylogenetic diversity. This idea behind phylogenetic diversity underlies all of the other measures that have been devised over time. I also wanted to point out this really unique tool called the OneZoom tool. You can find it online, and I will show you a link to this particular website. And it is a way for you to explore biological diversity. But what is really interesting is that the tree is built not just based on our current knowledge of phylogeny, but if you look at the different colored leaves on the tree itself, those leaves are actually indicative of the IUCN categories that we find in the red list. So I'm going to show you really quickly by going online what this OneZoom website lets you look at. And so here's the OneZoom website, and I've already kind of narrowed in on a particular group of amphibians, frogs, which we know are under severe threat from extinction. And you can zoom in on this tree wherever you would like to get into some of the smaller branches. You can also zoom out, and you can hover over this little navigator tool, and it will take you to one of the more recent ancestors of a larger taxonomic group. So if I wanted to just look at all frogs and toads, I could actually zoom out and do that. So that's a really, really neat tool. So it takes us all the way back to the ancestor of all frogs and toads right here. So what you all will notice is that we have branches that are just gray, but if we zoom in on particular areas of the phylogeny, let's just go to this branch, which looks really scary, and this is actually the one that is in the PowerPoint. You will notice that there are these red leaves, or taxa, and then there are green ones. If you look at the red ones, so if we zoom in on this Gardner's Seychelles frog, you will notice that its conservation status is endangered. Here right next to it is another one, the Seychelles palm frog. The Seychelles is a small island off of the eastern coast of Africa, and it's located very close to Madagascar. You will notice that here's another species that is critically endangered. So any of the ones that are vulnerable or threatened in some way are labeled here. Here's one that's endangered. This is just the Seychelles frog. If you look at some of the green leaves, you will notice that this particular species, this is the eastern ghost frog, is of least concern, according to the IUCN Red List. So what this website does is it provides you with information not just on the evolutionary relationships between these species, but their red list status. And that is just fantastic information to have because it allows us to start thinking about phylogenetic relationships in terms of conservation. So here's a question I have for you, and we can talk about this in our group chat. Let's say that we decided, I'm going to zoom out a little bit on this. Let's say that we decided to focus our conservation efforts on, let's zoom out a little bit more even. Here's our tetrapods, and here's the amniotes in this part of the zoom tree. Let's say that we focused on this little marsupial species. So I'm going to zoom in on this little guy. I happen to have a strong fondness for marsupials. And let's zoom in here on the close to the tip of this tree of the marsupials. And I am going to zoom in on this little leaf right here, the quokka. So let's check it out. The quokka is this cute little marsupial that is vulnerable. Okay, so it's not yet endangered or critically endangered, but let's say that I wanted to focus conservation efforts on this species. It's found in a very small area. I know that it's endemic, and the question would be, I'm going to zoom back out, should the quokka be a focus of conservation? Now you might say, well, compared to what? That's a good question. Should the quokka be a focus of conservation along this branch? And one argument might be yes. The quokka, or maybe another species close to it on this tree. Let's click on this other red leaf and see what it is. This is the bridled gnat-tail wallaby, is also vulnerable. Here's a wallaby that's perfectly fine. That's interesting. But let's say that we decided that, yes, we really like the quokka. It is a little charismatic species. People really like it. And maybe it's used for food or something. It's got some medicinal component. Maybe we use something from it or harvest something from it for human use or human health purposes. Maybe we found an antidote to a venom, or maybe we found the cure to the COVID-19 disease. So why would you want to conserve the quokka, or why would you want to focus conservation efforts there? And if you think about it, if you zoom farther and farther out on this phylogeny, and the quokka is now hidden over here behind this picture, if you were to focus conservation there, what you're doing is you are literally preserving or conserving this entire branch that captures the marsupials. This entire branch from the ancestor, which is the Therians, and then if we follow this branch along, down the marsupial line to this small group here that I zoomed in at the very end here, we are literally conserving that many millions of years of evolutionary time. And in fact, you can even look at how long ago these critters evolved. Marsupials evolved 87 million years ago. If you take that and you try to focus on the quokka, and you look at the time here, there was the quokka. The quokka evolved 10.6 million years ago. you are preserving about 70 million years of evolutionary uniqueness. That's pretty interesting. So this is just a different way to look at conservation. Instead of just looking at preserving or conserving some of the most charismatic or sexy species that people really like, such as, say, the koala, right? Let's look at some of these other species and look at how much phylogenetic diversity we can actually preserve or maintain on our planet. The one thing about this is that we don't know what's going to happen in the future, right? It could be that in the future, the quokka does perfectly fine based on new data that we collect. And we might have put all of our focus on the quokka when we should have been focusing on another closely related species instead, right? So this kind of gives you an idea of how phylogenetic information can be useful. So we'll come back to this concept of the IUCN Red List once again in a little while. So I encourage you to go look at the OneZoom website. All right, I will post the link on our Moodle page. Let's talk about another measurement that is used. This is called the taxic diversity measure, and it was proposed in 1991 by Vane-Wright. And basically what this is it's a measure that allows us to determine which area or region among several should have preservation or conservation priority. So here we're still using phylogenies, but there's some additional information built into that phylogeny. All of the following measures that I will introduce, and there's really only one more after this, are based on the phylogenetic diversity measurement or phylogenetic metric idea that was proposed in 1992. So what I want you all to imagine here is that we have a phylogeny of species A through E, and what these values represent, we've got W, which is a weight score. That weight score really is just a measurement, and this is assigned beforehand, a measurement of its some kind of value. The value could be the branch length, right? So A and B have short branch lengths. D has a branch length that's actually more than twice as long as A and B, so these values aren't necessarily branch lengths, but perhaps they're a measure of uniqueness, okay? So of evolutionary distinctness. So these weight values, I would have to give these to you. Those aren't ones that you can make up. But you can imagine that the shorter the branches, the less unique that particular species is in terms of its built-up feature diversity over time. Shorter branches means less evolutionary diversity distinctiveness, whereas the longer branches mean a lot more time and potentially a lot more unique mutations that result in new features, feature diversity. So that's the W, is the weight score. And R1, R2, and R3 are regions. So these three values, or three columns, are regions. And let's say that before we do this, we go into this study trying to determine which of three different distinct ecological areas are the most phylogenetic diverse and perhaps the most worthy of conservation efforts. In each area, we've got where the different species live. In each area, we have a red dot that suggests that those species, A, B, C, D, and E, live there. So in region 1, we find species A, B, and C. In region 2, we find B, C, and D. And in region 3, we find C, D, and E. There are, if you notice, 3 species found in each of these regions, so they are equally species rich. They are equally species rich. They have the same number of species. And this is a highly simplified example, right? Because 3 really isn't a lot. Equally species rich. And what we want to do is try to estimate, okay, which of these regions should we focus our conservation efforts on? Which of these 3 areas should be conserved or preserved based on these species? Well, If you are strictly using the number of species or taxonomic diversity in this area, which we talked about at the very beginning, how the heck would you make that decision? You couldn't, because they have the same number of species, and this is where phylogenetic information becomes really useful. Okay, so these measures on the bottom, the T here is the total. And what this score here is, the 9.3, is a total of all the weights, of all the weight possibilities. So it's 1 plus 1 plus 1 plus 3 plus 2 plus 4. That gives us 9.3. And then what you have here is the total score for region 1, region 2, and region 3. If you add up region 1 scores, these weight values or those uniqueness values, you've got 1 plus 1 plus 1 plus 3. These values really we can just call taxic diversity values, okay? Because this is the taxic diversity scoring mechanism. So we've got 1 plus 1 plus 1 plus 3, which gives us a total of 3.3. If we take region 2, we take 1 plus 1 plus 3 plus 2, which gives us 4.3. And then finally, for region 3, we take 1.3, 2, and 4, and we get a total taxic diversity score of 7.3. You will notice that region 3 has the highest diversity score. So that should give you an idea of where we are going with this measure. The first thing that we're going to calculate is P1 and P2. So we've got our total weight scores or our taxic diversity scores, but what we're now going to calculate is a value P1, which is called the percent diversity for each region, or the proportional diversity for each region. So what you do for this one is, let's say for region 1, what you're going to do is take your total taxic diversity score, which was a 3.3, and you divide it by the total taxic diversity for the tree, which is 9.3. And what that gives you is 0.35. or 35%. For region 2, if you do the same thing, you take 4.3 divided by your total score of 9.3, and you end up with 0.46, or 46%. If you do the same for region 3, you're going to end up with 7.3 divided by 9.3, which gives us 0.78 or 78%. So it's a proportion of how diverse that region is relative to the total taxic diversity in all three regions that you are comparing. And what you will notice, of course, and we could have determined this or predicted this just based on the taxic diversity scores, is that region 3 has the highest taxic diversity measure, which also equates to the highest percent diversity for P1. P1, if we think about it, or if we define this, is the priority number 1. It is the area that gets the top priority for conservation. So we already know that region 3 has the highest priority value. 78% of the taxic diversity of all of the diversity in this phylogeny is found in region 3. It has the highest priority score. Next, we're going to calculate P2, which is the region that gets second highest priority after region 1. So P2 is the percent diversity for taxa unique to a region when compared to the most diverse region. So if we compare region 1 to region 3, which we've already assessed has the highest priority score, what species are unique to region 1? And if we look at this, species A and B are found in region 1 but not in region 3. So what we do is we add up their weight scores, which would be 2, and we divide it by the total taxic diversity score for the entire tree and all of the taxa. And what that does is it gives us a score of 0.22 rounded up, which gives us 22%. Similarly, if we do the same thing with region 2 and we compare it to region 3, what species are unique there? The only species that is found only in region 2 but not in region 3 is going to be species B, which gives us a score of 1 out of 9.3, which is the total taxic diversity score. And that gives us a value rounded to about 11%. And what this suggests then is that region 1 should get the second highest priority score. So region 3 gives us 78% of the taxic diversity. Region 1 gives us 22%. When you are calculating these, you have to make sure that you have worked through all of your different priority regions. It is possible that you might have 4 regions and you need to find the top 3. So it depends on what you are trying to do and how strict you are in your assessments. So I hope this gives you a general idea of what these P1 and P2 values mean. One thing that you will realize is that the final taxic diversity scores for region 1 and region 3, which are 22 and 78, they equal 100 or 100%. What this means is that the analysis is now finished. We have actually accounted, just by looking at region 3 and 1, we have accounted for 100% of all the possible taxa that are found in this tree. Just by conserving or preserving areas region 1 and region 3, we are actually conserving 100% of our species. And here's a final question for you. Make sure that you can answer this. Which region 1, 2, or 3 has the highest conservation priority? Make sure that you can work through this problem. Which has the second highest, and which has the lowest. So this is the Vane-Wright taxic diversity measure that helps us establish how we might make decisions about areas instead of just species. And one nice thing about focusing on areas, as you can tell, is that if you can capture an area and its diversity, you don't have to worry about just one species. You can actually focus on a variety of them. Next, we're going to look at the edge species metric, and this is the very last one that I want to introduce you to. So what we're going to be doing here is considering the extinction risk of species along with our evolutionary distinctiveness. And so this metric is called EDGE, and EDGE stands for Evolutionary Distinct and Globally Endangered Existence metric. So we're going to be considering both the distinctiveness of each of those species in the phylogeny and the currently known red list category to which that species belongs. That is our EDGE metric. And how this works is we're basically going to measure species' relative contributions to phylogenetic diversity, and that's the evolutionary distinctiveness measure, but we're also going to consider their status. So we're going to measure evolutionary distinctiveness first, which I will do here, and then we're going to talk about the extinction risk. One thing that makes this metric a little bit more tricky is that for a lot of species, we don't have their extinction risk measures yet. They haven't been assessed, especially if we think of a lot of insects and other things. So we've really just used these EDGE metrics for mammal groups or other larger vertebrate groups. And we're going to calculate our evolutionary distinctiveness score first. So what we're going to be doing is looking for our above branch scores, which is the branch length of just that species. And then we're also going to be looking at the number of species subtended by branches for our scores below the branch. What that basically means is that you include a score of the number of species that are captured by that stem branch. So let's walk through this and we'll be able to understand what all this means. So I'm going to put all of our above branch scores first, which is our branch length scores. One thing that helps here is that there is a scale on the bottom, and our branch lengths are measured in million years before present. For most of these EDGE analyses that are being performed today, we actually have to have knowledge about how long these species have been around. So whenever I give you a problem that you have to work through regarding the evolutionary distinctiveness problem, you're going to be given a scale to work with. So first we're going to calculate above branch lengths, and based on our scale below, this branch length is 1. So the branch length just going to A is 1. And the one going toward B, just the branch that B is on, is also a 1. C, if we look at the scale, is a branch length of 2. D is also a 1, and so is E, and F and G are 0.5. Now let's go further back. What is the branch length here that subtends A and B? It happens to be a 1. What about the branch length here that subtends A, B, and C? That happens to be a 1. And how about this branch that subtends A, B, C, D, and E? This happens to be a 2. So we're just looking at branch lengths from the branch start point to where it splits and where there is a node. All right, the branch length here that goes toward F and G, the branch that subtends those two taxa, are going to be 4.5. Hopefully that's done correctly, right? So what we're going to do next is calculate the below branch scores, just so that you can understand how these work, and I'll do this in blue. So what we're going to calculate here is the number of taxa that are captured by that branch. Here, we're just looking at a branch leading to A, so our below branch score is a 1. Here, below the branch, there are 2 taxa captured by this stem branch, so that gives us a score of 2. Here, we have a branch that leads to species A, B, and C, and so that would be 3. Here, we have a branch that leads us to A, B, C, D, and E. That gives us a score of 5. We've forgotten to put scores below B and C, but we've got 1 and 1. For D and E, we've got 1 and 1 separately. And this score below is a 2. Oh, and you will notice that I forgot to put a score above here. What would this score be? What would this score be above here? Very good. That would be a 2. Okay, let me quickly fill that in. So this would be a 2 that I forgot to put earlier. Okay, and then finally the scores below these bottom branches. What would be the number below the F branch? This would be 1 because there's just 1 species captured there, and a 1 below the G. And then finally, here, this would be a 2. So now that we've calculated our ED scores for each of our species, what we do with these scores, those scores both from above and below each branch, is we are going to add those values together, and you can think of them as fractions. So we are basically going to sum together the values for each species, based on the values that both lie above and below each of the branches. And so what we're going to do is, as an example, we are going to take, so here's an example for species 1, or A. Example for species A is we're going to take 1 divided by 1, which gives us this first branch fraction or ratio, plus 1 over 2, which gives us this second ratio, plus 1 over 3, plus 2 over 5. And you do that for each of your species. You're calculating the distinctiveness, which is a measure of the length of each branch that leads to that species over the length of the branches which capture all of the species within that clade. So it's giving you a measurement or a measure of how unique or distinct that species is relative to the clade. So finally, you end up with values that look like this. You can double check the math here, but species A should end up with 2.23 and so on and so forth. And then I have this question on the slide here. Higher ED scores usually suggest higher conservation priority. Without knowing anything about the risk assessments for these species, why would that be? Why would a higher ED score suggest higher conservation priority? So, knowing what you've learned now about phylogenetic distinctiveness and phylogenetic diversity, make sure that you can answer this question. So spend a little bit of time. I encourage you to pause, and we're going to talk finally about the second part of this measure, which is the risk assessment. And what we're going to do now is combine that ED score with the extinction risk for that species. In order to calculate this, everyone, you would have to have the assessment scores that the IUCN have gathered from the red list in order to get to the final value. So the edge score, the final edge score, is equal to the natural log of 1 plus the ED score, the evolutionary distinctiveness score, and then you add that to the GE score, which I'll define in a moment, and multiply that by a natural log of 2. The GE score is the red list category, so least concern would be 1, not threatened would be 1, vulnerable would be a 2, endangered would be a 3, and critically endangered would be a 4. If a species is extinct, which is what the last two categories are on the red list, remember they're extinct already, so there is really no reason to focus conservation efforts there. So this is the calculation you would use, and you can kind of practice these numbers and these values, but the thing is here that you don't know what these species are, you don't know what A, B, C, D, E, F, and G are. I would have to give you that information. And what you might have to do on the exam is go and look up some of these species in order to answer this question. So be ready for something like this and come with any questions that you have. Finally, I wanted to show you some of the results from one of the very first EDGE papers, which was published in 2007. I will also post this paper for you to take a peek at. I know that there are a lot of papers and documents that you're going to have to sift through for this topic, but it's just because there's so much cool and unique information that relate to conservation biology, which makes me kind of sad because we can't really delve into all the details. But what I wanted to show you is the study which calculated the EDGE scores for a bunch of different taxa, and they focus mostly on mammals because really mammals and some vertebrate groups are the only ones that we have a lot of EDGE score information for. And what that means is that the phylogeny is established and we have risk assessments from the red list for these species. If you had to do this for invertebrates, you would be SOL. We don't have clear phylogenies for most invertebrates, and we don't have risk assessments done for most invertebrates. So our data are biased because we've really only been able to look at these large charismatic species that we know. But what you will notice in this figure is that the EDGE scores, which range from say 0 to 6, and remember the EDGE score includes both the evolutionary distinctiveness as well as the red list extinction factor or score. What we will notice here is that the higher you go on your EDGE scores, the higher the EDGE scores get in these species, the more likely those species are to belong to those higher risk categories. So green would be least concern, for example, or there's not enough data, or there's enough information to say that those are not threatened. And then the red category would be critically endangered, the orange would be endangered, and then the yellow would be vulnerable. So those red, orange, and red are our threatened categories. So this kind of makes sense, that the species that tend to be in those higher categories in terms of risk also tend to have higher edge scores. But there was something unique that this paper found. And so here's a list of the different taxa that made the top 100 edge species. So the species that had a combined edge score of something closer to 6. And I'm listing them here. We've got both the giant and the lesser panda. The giant panda is the black and white panda we know, but the lesser panda is that really cool panda that looks more like a raccoon than anything else. In fact, it is a relative of a raccoon, and it is not a panda bear at all. We've got the orangutan, which always makes this list. We've got 2 elephants, which are both the African and Asian elephants, 4 different rhinoceros species, 2 tapir species, 2 baleen whales, the dugong, and then a manatee. There are multiple manatee species, but one of them made the list. So these made the top 100 edge score list, but what was also found was that there were 46 amongst the 100 other species that are small, obscure, lesser well-known species that made the 100 edge list. These are things like small mice or shrews or things that most people don't really pay attention to because they're not charismatic animals. And so the lesson here is that we should really be considering the risk factors, the extinction risk of species when we're making these decisions. Yes, it is very easy to convince the public and other stakeholders that we should invest more money and more resources into conserving the giant panda because it's a sexy, cute, fuzzy animal. If you compare that to a little obscure shrew from Southeast Asia, that becomes really, really difficult to sell. And other findings from this particular paper was that 37 of the top 100 edge species found in the edge analysis are not really strict and endemic species. What that means is that they live in areas where their populations might be declining, but they're not defined as endemics. So since there's always a focus on endemic species, this paper has actually suggested that instead of focusing on endemics, we should focus on species that are in these higher risk categories rather than endemics, because 37 of the top 100 species were not endemic species. They are species that have population ranges that are, or population sizes that are shrinking, but their geographic ranges are fairly large. So they aren't really considered endemics. So should we focus on endemics purely? Should we focus on hotspots entirely? Maybe not. We should be considering these risk assessments. Finally, as the last closing slide, which I'm sure you're so excited about, because this is the last slide of this topic, what should we conserve and preserve? What kinds of places, what kinds of species? I've shown you a lot of different examples of things that we should focus on and areas where we should invest more resources or potentially more time, more research. What do you think should we conserve and preserve? What kinds of areas? What kinds of animals? And we're not talking just animals, right? We're thinking about plants too. So think about that. I'm going to leave you with this blank slide and we will discuss this as a group. We are done, everybody, with this topic. Thank you for enjoying this with me.